A very good morning to you all, and welcome to our fourth lecture of the online lecture series on techniques in plant molecular biology. So, in the last three classes, you have learned about the theoretical aspects of gene cloning, and today we will learn how to practically clone a gene. So, before we start into the practical aspects, I want to tell you about two important cloning systems that are very commonly used in all labs. So in most of the molecular biology lab, cloning of a gene starts with a TA cloning step. So what is a TA cloning? A TA cloning vector is a plasmid vector which is available commercially in a linearized form. And on the both ends of the linearized vector, we have a single T overhang over here. So on both the ends, at the 3' end, we have a single T overhang. And on the other hand, when we PCR amplify our gene of interest or the fragment of DNA of our interest, the Taq polymerase adds one single A overhang at the 3' ends. So automatically, we have our PCR product with a single A overhang on at both the ends. And this addition of the deoxyadenosine residue is independent of the template. So whatever template you are using, whatever primer you are using, the single deoxyadenosine overhang at the 3' end of the amplified fragment is common. So naturally, we have our PCR product with single A overhang, and we have the linearized TA vector which has a single T overhang. And now if we incubate this PCR product and the linearized vector, and if we incubate them in a proper buffer system and add some DNA ligase to it, then this A and T overhangs will base pair because they're complementary to each other, and then the ligase will form a phosphodiester bond here. So of course we will have our PCR product ligated into the PGMT, the TA vector system. So this is the place where our PCR product gets ligated between these two T overhangs. Now this TA vector is normally commercially available from Promega, and they call the system PGMT easy vector system. Another additional benefit that PGMT easy vector provides is there is a lot of option of restriction sites present on both sides of the insert. So once we have our PCR product insert ligated inside this linearized plasmid, we will have an option of many restriction sites on both sides of the inserted fragment. So even if we do not have any restriction sites added to our plasmid, we can choose from these restriction sites to cut the fragment from this vector and to subclone it into our final destination vector. Now this is a common a reaction component that we generally use, and this is in this we add the proper ligation buffer, then we add the PGMT easy linearized vector, our insert, that is the PCR amplified product, of course the T4DNA ligase for ligating them together, and then we volume make up the reaction mixture with nucleus free water. And if we incubate it for around 60 minutes, then we will transform this ligation product into the E. coli cells, competent E. coli cells, and of course we will screen the transformed cells on antibiotic plate. And in case of PGMT easy vector, we have an ampicillin selection because the vector has ampicillin resistance gene. And that is how we can very easily clone our PCR product, our gene of interest, into the TA vector. And in most of the molecular biology lab, this is essentially the first step in gene cloning. And after we clone it inside the TA vector, it becomes easier for us to sequence the gene of interest, whether we have amplified the correct sequence or not. And this step is very crucial because if we don't amplify the correct product, even if we clone it further into our destination vector and introduce it into our host cell, we won't get the correct gene product. So this is a very crucial step. Now another variation of this TA cloning is the topo TA cloning, and this topo TA cloning is marketed by Invitrogen. This is essentially the same thing. The vector is linearized and it has a single T overhang at both the ends. But in addition to this, they have one topoisomerase 1 covalently linked to this T overhang. And in this case, we do not need to add any extra DNA ligase in the reaction mixture. Once the T overhang base pairs with the A overhang of the PCR product, this topoisomerase 1 catalyzes the ligation of this insert and the vector. So we don't need to add any DNA ligase from outside. The topoisomerase 1 that is ligated covalently with the bonded covalently with the vector catalyzes the ligation reaction. So it is essentially the same reaction, but in case of PGMT-EZ, T4 DNA ligase catalyzes the phosphodiester bond formation, and in case of topo-TA cloning, topoisomerase 1 catalyzes this phosphodiester bond formation. Okay, so in most of the labs, this TA or topo-TA cloning is essentially the first step in case of cloning a particular gene. Now let us come to a practical example. So before cloning a gene by ourselves, let us first have a look what are the essential steps that we normally follow for cloning a gene with known sequence. So the first step is of course to retrieve your gene sequence from a database, say for example from NCBI database, and then to select a suitable cloning vector for my gene of interest. And this depends on the size of the vector as well as the motto why we want to clone the gene, whether we have to clone a gene or a promoter, we want to ligate it with a reporter gene, we want to introduce it into a plant system, or we want an expression system in yeast or E. coli. So according to our need, we have to select a suitable vector. Now once we have selected our vector, it is now important to select a pair of restriction enzymes which we will going to use for cloning our gene. So we have to select 2 restriction enzymes for which sites are available in the MCS region of our vector at a proper site, and also the sites are not present inside our gene of interest. This is important because if the sites are present inside the gene of interest, when we will try to digest our construct, our gene of interest will get chopped off, and this will lead to truncated gene product. That means our gene of interest will get destroyed. So the selected restriction sites must not be present within our gene of interest, but it should be present at a proper site inside the MCS of the cloning vector. Next, once we have selected the RE sites, now it's time to design the gene specific primer. Primer designing can be done manually or through software. For example, we can use Primer 3 software, which is very widely used. And after we design the gene specific primer, we have to add the RE sites, the restriction sites, in the 5' end of both forward and reverse primer. This is essential. We have to add the RE sites at the 5' end of the forward and reverse primer. Now, once we have designed the primers, we will PCR amplify the gene of interest. Now, here we have to remember that how to amplify the gene of interest. In most cases, we will amplify the gene of interest from RNA. That means we have to isolate RNA, then we have to synthesize complementary DNA or cDNA from it, and then use the cDNA as template and amplify the gene of interest using gene specific primer. But if we are cloning a promoter fragment, of course, we have to use genomic DNA for that. Next, the PCR product will be run on agarose gel, and then we will purify our desired band from the gel using any commercially available gel purification kit. Now, once we have purified the our gene of interest, that means the desired insert DNA from gel, we have to quantify the amount that we have. And next, we will move directly into TA cloning as we have seen just now. After TA cloning, the ligation mixture will be transformed into E. coli cells, and then the E. coli cells will be screened on antibiotic plates to select the positive clones, that means the positive E. coli colonies that harbor our construct. Now, once we have cloned our fragment into a TF vector, we will of course check the sequence through PCR using gene-specific primers, then restriction digestion using the two RE sites that we have used, and finally we will sequence the gene product so as to be confirmed that we have amplified the correct sequence. Once all this has been done, now it's time to subclone our fragment into the destination vector. So for that, we will double digest the TA clone on one hand and the selected destination vector on the other hand. And we will double digest both the vectors using the selected RE pairs. Then we will run this digested product on agarose gel along with the ladder, and then we will gel purify the digested insert and the digested vector. Once we have isolated and purified the insert and the digested vector, now we will ligate them together and using T4DNA ligase, and then transform the ligation product into competent E. coli cells. Then similarly, the positive E. coli clones will be screened on antibiotic plates, and then we will reconfirm the clone through PCR, RE digestion, and maybe through sequencing. So these are the steps that we should follow for cloning a gene in our laboratory. Now let's have a look at a practical example. So essentially, the first step to clone a gene is to retrieve the cDNA sequence from the database. So in this case, we want to clone this gene which has the accession number XM015776394. So now we have to retrieve the sequence from NCBI database. So for that, we will move to the browser and just we will go to NCBI and we will search in the nucleotide library. We will paste the accession number here and retrieve the sequence. Now, while retrieving the sequence, we must keep in mind that we are going to clone a gene. And in most eukaryotes, we have both introns and exons. So, while cloning a gene, we want to minimize the size of the fragment without leaving out any essential part. So, we can safely remove the introns. For that, we want to select only the cDNA sequence. So, for retrieving the cDNA sequence specifically, we have to click on the CDS over here and then on FASTA sequence here. So, this will retrieve the FASTA sequence of only the CDS part of our gene. And selecting the CDS region only will essentially minimize the fragment size. So, now we can copy this sequence and paste in our library. So, that's how. Yes. So let's go back to the slides. So we have retrieved our sequence. So this is the cDNA sequence we have used, and I have already told you why we have taken cDNA, because we don't want to get the introns in our sequence. So we will use only the exonic part, which is available only in the cDNA sequence. Next, after we have retrieved the sequence, we have to determine what is the size of our gene. In this case, the size of our gene is 1 kb, which is much less than that of 10 kb, and so we can use safely a plasmid for vector. So among the plasmid, in this case, our motto is to clone this gene into a binary vector, because we want to introduce it into a plant system. And we also need to tag this particular protein, this particular gene with the GFP, so that we can very easily visualize the expression of the gene. So once we have introduced this fragment into the plant cell, we can easily visualize the expression pattern of the protein, both in the tissue specific manner as well as in the subcellular localization. So we need a GFP which will be tagged with our gene of interest. Now for plants, the very commonly used plasmids are PBI121 and a Picambia series, including Picambia 1303, 1304 and 2101. There are many more as well. And we have a payget vector. So how these vectors look like? Let's see. This is the PBI121 vector, and you can see that this PBI vector has, this is the expression cassette which is to be expressed inside the plant cell. This is the 35S promoter, which is a constitutive promoter. That means it will be expressed, the gene will be expressed in all the tissues and under all conditions. So this is the promoter, and here we have a GAS gene, and then here is the NOS terminator, and these are the options of RE sites available for cloning. But we do not have a GFP over here, so we'll leave out this plasmid. The next one is the Picambia 1304, and see here we have a 35S CAMB promoter over here. This is the MCS, and then we have MGFP and fused GAS, and here is the terminator. In this case, we have a lot of options for MCS over here. That means before and after the promoter, CAMB promoter. So this vector is very useful for cloning of promoter sequences, because we can replace the 35S promoter and put our promoter over here, and the promoter will direct the expression of GAS and GFP genes. But in this case, this vector will not be very useful for our purpose. And the last one is the payget vector. Here also we have a 35S promoter, which is a constitutive promoter. Then we have EGFP, and then we have MCS region or the cDNA cloning polylinker. This polylinker has 4 options of RE sites: EcoR1, SMA1, HIN3 and BAMH1. Apparently this vector appears to be useful for us because we want to tag our gene of interest with a GFP, and we want to place it under 35S constitutive promoter. So now it's time to check whether these RE sites are present inside our gene of interest or not. So the next step is to select RE sites and design the primers. These RE sites can be checked by using NEP cutter, and then we can virtually digest our gene of interest and our selected plasmid by using restriction cutter 3. So again, let's go back and check with it. Let's go back to the browser. First, we will go to NEP cutter. This is NEP cutter version 2.0. Here is the place where we have to paste our sequence. So we have already retrieved our cDNA sequence. We will just copy and paste it over here. Remember to choose whether your sequence is linear or circular. In case of gene of interest, it is linear because all eukaryotic genes are linear. So we have selected linear over here, and then we will click on submit. So here is the result. Now let's look at what are the available restriction sites present inside our gene of interest. And interestingly, we do not have EcoR1 and BAMH1 sites that we have already selected. So because these 2 sites are not present inside our gene of interest, we can safely select these 2 RE sites. So to reconfirm, we will again go to the restriction cutter. Restriction cutter 3. I'm sorry, restriction mapper 3. And this is the page. Now we will again paste our sequence over here, and we will select our genes, our restriction enzymes. We have BAM-H1 and the EcoR1. Again, remember to correctly select the conformation. In this case, we have a linear one. Then we will click on virtual digest. This is the results we get. We don't have any restriction site for BAM-H1 and EcoR1 within our gene of interest. Now the next part is that we have to check the sequence of our vector. So we will retrieve the sequence for PEGAT from this NCBI server. This is the cloning vector PEGAT complete sequence, and so here also we need the FASTA format. We'll click on FASTA and retrieve the sequence. This is the sequence for our vector. We'll copy it and paste over here. But in this case, it is a circular plasmid, so we will select circular. We have already selected the RE sites. Now we will click on virtual digest. See, it's very interesting. If we use the enzymes BAM-H1 and EcoR1, we will get one fragment of length of 12570 base pair and a second fragment of 24 base pair. That means this fragment will come out of our vector and there is no additional site. So we can safely select this RE pair. So let's get back to the slides. Yes, so we have selected our restriction sites. These two are the so we can select this EcoR1 and BAM-H1 RE sites. So this is how our NAP site looks like. We have already seen, and this is how the restriction mapper tree looks like. So now as we have selected our restriction sites, it's time to design primers. Remember, primers can be designed using softwares as well, but in case of cloning the full length fragment, it's always advisable to clone it, to design it manually. So how to design the primer? Remember, we need one forward primer and a reverse primer. Designing forward primer is very easy. We will just select a part of the sequence from here, from the start ATG of the sequence, and then we will calculate what is the approximate TM or melting temperature. How to calculate TM? Now this is the part that we have selected, the highlighted part. Then we will count how many A is there, how many T is there, how many G is there, and how many C is there. So the easy formula is this: A plus T multiplied by 2, and then we have to multiply by 4 into G plus C. This gives us the TM. So the TM is A plus T multiplied by 2 and G plus C multiplied by 4. So we have to count the how many A, T are there. So we have 7 A plus T, so 7 into 2, and the rest 12 are G plus C. So 12 into 4, so total we have 62. So our forward primer will be this one. Now remember, we have selected the RE sites. What are the RE sites? Let's go back to that slide. This is the EcoR1. So we have to place the gene in such a way that the ATG lies after EcoR1 and the terminator lies just before BAMH1. So we have to add EcoR1 site on the 5' end of our forward primer. So it's very easy. You can find out the restriction site for EcoR1. This is the restriction site for EcoR1. So we have added this over here. We have added some overhang over here. This is sequence non-specific, just to provide some space for the enzyme, restriction enzyme, to sit over here. And then we have put the sequence of our forward primer. So our final forward primer will be 5', this restriction sequence, and then our primer sequence. And remember, annealing temperature in the PCR will be approximately Tm minus 5. So if this is 62, our annealing temperature will be 57. And annealing temperature should always be above 55 degrees centigrade. So we have to adjust the primer length accordingly. Now in case of reverse primer, remember we have to take something which is complementary to this one, because it will bind on the complementary strand. So we have selected this highlighted part as the sequence, and then we have calculated the Tm value. It's best to keep both the Tm values same. So we have to adjust the length of the primer accordingly. So these are the 80. We have to count the number. The total we have 9A plus T, so 9 into 2, and 11G plus T, so 11 into 4. Together we again have 62, which exactly matches with the forward primer. So we can move on with this one. And in this case, we have to add BAMH1 site. So the final sequence will be, in the final sequence, we have to add the BAMH1 site on the 5' end. But in case of reverse primer, one important thing is that we have to take reverse complement of this sequence, because this reverse primer will bind to the reverse strand. So we have two strands of DNA. One will bind to the forward strand, plus strand, and the other will pair with the minus strand. So we have to take reverse complement. It's very easy to calculate the reverse complement. I'll show you. Just a minute. We will go back to the browser. Yes. Now we will type reverse complement. It's very easy. Just paste your sequence over here and click on reverse complement. They have already given this. So we can copy this and take to our slide. Yes. So we have taken the reverse complement. So the final primer will be 5', then the RE site, and then the reverse complementary sequence, and then 5', because we always have to write it in the 5' to 3' direction. So now we have our primers ready for cloning. Now once we have our primers ready, the next step is to PCR amplify the gene of interest. Now remember, because we are using a gene, we will always start with isolating RNA. Now, where from we will isolate the RNA? Of course, our tissue of interest. We have to keep in mind that whether our gene of interest is a constitutive gene or an inducible gene. If it's an inducible gene, we have to keep in mind in which tissue the gene expresses. The expression is maximum. Say, for example, there are some proteins which are inducible under stress condition, or it is present only in the leaf, or only in the root, or only in the inflorescence. So we will select that tissue where the expression of my gene of interest is maximum. So we have to select a suitable tissue and then isolate total RNA from that material. Once we have isolated the RNA, we will quantify the total RNA, and then we will use it to synthesize cDNA. cDNA synthesis kits, there are a lot of options and we have to select any one from them, and then we have to critically follow the manufacturer's instructions. So following the instructions, we can very easily synthesize cDNA, and then we will use this cDNA to PCR amplify the gene of interest using our designed primers. That means the gene specific primers along with the RE site. Once the PCR is ready, we will run the PCR product in an agarose gel, and this is how we should load the gel. In the first lane, we will load a suitable DNA ladder. So because our gene of interest has a size of 1 kb, we will of course use 1 kb DNA ladder, and in the second lane, we will run the PCR product. This will ensure that we have amplified the correct gene product. We can check with the size of the DNA ladder. Once we have amplified the correct gene product, we will purify the gene product, the PCR product, from we will purify the PCR product from the agarose gel, and this can be easily done by using any gel purification kits. A number of options are present commercially. So now, after we have purified the PCR product from the gel, we will quantify it, and then we will directly move on to the TA cloning. Now, here we have an important step. We have to calculate the molar ratio of the insert is to vector before we start with the ligation. Now, how to calculate the insert is to vector molar ratio? Here we have a very standard formula. See, the nanogram of vector multiplied by kb size of insert divided by kb size of vector, and then multiply it with the insert is to vector molar ratio, and that will give you the nanogram of insert. That means how much insert you have to add to your ligation mixture. Now, in case of PGMT, because we will first do the TA cloning, in case of PGMT, the manufacturer has provided us with the linearized vector, and the concentration is 50 nanogram of vector per microliter, and they have suggested us to take 50 nanogram of vector per reaction. So this one is 50 nanogram according to their instruction. In our case, the insert size, that means the gene size, is 1 kb. So here we will write 1, and the vector size in case of PGMT easy vector is 3 kb. So we have 50 nanogram into 1 kb divided by 3 kb, and they have suggested the ratio 3:1, insert 3 vector 1. So we will multiply it with 3 divided by 1. So that gives us 50 nanogram of insert. So we will set the ligation reaction according to that reaction that I have shown you on the first slide. Let's go back to it. We'll go back to that slide. So we will set the reaction mixture according to this reaction. So we will add ligation buffer. We will add the vector 1 microliter PCR product. This will vary. You have to calculate the how much insert you have. Then you have to calculate that how many microliter of your purified insert contains 50 nanogram of insert. You have to add that. Then you have to add T4 DNA ligase and volume makeup up to 10 microliter using nucleus-free water. All right. So we can very easily clone the fragment into the TA vector, and so after we have calculated the molar ratio and the amount of insert that is required, we have to do the TA cloning as per the instructions provided in the manual, and then we will transform the ligation reaction mixture into the E. coli cells, competent E. coli cells, screen the cells on antibiotic plate. In this case, we have ampicillin resistance, and finally we will confirm the positive colonies by PCR and restriction digestion and maintain the culture. Now here it is important to sequence the TA clone because we want to make sure that we have amplified the correct sequence. Otherwise, even if we introduce it into our destination vector and transform it into plant cells, the correct gene product will not be produced. So we will have truncated protein or a completely wrong protein product. So here it is very important to sequence and check that we have cloned the correct fragment. All right. So once we have completed this screening, we will maintain our clone in a glycerol stock for long time preservation. So now we have to subclone this insert DNA from the TA clone into our selected payload vector, which is our final vector. So for that, we have to now double digest our TA clone and our vectors simultaneously with the selected RE pairs. So in our case, we have EcoR1 and BAMH1. Okay. So in this case, after digestion, we will run agarose gel, and this is how we should run it. We will, in the lane 1, we will add a 1 kb DNA ladder. In the lane 2, we will add the digested TA clone, and in the lane 3, we will add the digested payload vector. And remember, while purifying the bands, we will take the smaller band, that means 1 kb band from TA clone, and the larger band, larger fragment from the vector, because we don't want the fragment that has come out from the vector. We want the rest of the vector. So we will now gel purify the insert DNA and the digested vector. Now once we have the purified digested vector and the TA clone and the insert DNA, we will again quantify them and proceed for ligation reaction. So in this case also we have to do a calculation similarly, and in this case the size of the vector will be 12.5 kb. The size of payload vector is 12.5 kb. So we will write 12.5 kb over there, and normally we use 100 nanogram of vector. So accordingly we have to find out, we have to calculate the size of the insert DNA, that means the amount of the insert DNA that we will use for the ligation reaction. The formula is same as this one. We have to again calculate the amount of insert to be taken using the same formula. Just we have to use the nanogram of vector should be 100. Then the size of the insert is same 1 kb. Size of the vector here should be 12.5. And the ratio can vary. It can vary from 3:1, 6:1, 1:4. You have to optimize, but we can, as a thumb rule, we can start from 3:1 ratio. So now we can easily set for the ligation reaction. After the ligation has been done, we will transform the ligation product into E. coli cells similarly and screen them on antibiotic plates. And finally, we will confirm the clone by PCR using gene-specific primer and restriction digestion using our restriction enzymes, that is the EcoR1 and BAMH1 in our case. So with that, we have already virtually cloned a particular gene. Now it's time for you all to check your knowledge. So this is the task I have for you all. Select a suitable, I'll be providing you with a sequence of this gene X, and that has to be expressed in a host cell. Proper expression should be there. So you have to put it in after a promoter and before a terminator sequence. Now you have to select a suitable vector from the options. That means we have PBI 1:1, Picambia series, and the payload vector. You have to select a suitable vector from them, and then you have to select proper RE sites for directional cloning, and then you have to design gene specific primer along with addition of RE sites in the primer. And finally, you have to calculate the amount of insert that is required for ligation. Given the molar ratio is 1:3, and the purified digested vector to be used is 100 nanogram. This is your task. So till then, thank you. We'll come back with a discussion with this task.